This is Syed Muktadir Al Sium from Bangladesh Agricultural University, and welcome to my presentation on ANTIFAM 6.0. Are we close to getting rid of spurious proteins in sequence databases? Let me start with some facts. As genome sequencing is becoming more common in today's world, many false or spurious genes are being predicted during protein sequence prediction using computational methods. Even if we presume the false positive rate of this method is around 1%, then within a database of 180 million proteins like UniProt, we'd still expect to find 1.8 million spurious protein predictions. Now, what are the consequences of these spurious proteins? Future genome projects will annotate new protein coding genes by similarity to the first spurious open reading frame. This can lead to entire families of spurious open reading frames. In the worst case scenario, these spurious families may even be annotated as having a function. For example, a spuriously predicted gene on the opposite strand of ribosomal RNA had been given the incorrect function of cell wall hydrolase. One existing tool that can aid in finding gene misprediction is the ANTIFAM database. ANTIFAM is a collection of profile hidden Markov models that can be used to identify members of potentially spurious protein families. However, till now the ANTIFAM approach does not scale well because identifying spurious protein is very slow and manual process. Each family requires the effort of a curator to build it and verify its status as spurious. So, only 72 families could be added after 5th release of ANTIFAM in almost 8 years. So, another machine learning based tool, the SPURIOUS, was developed to help identify spurious protein predictions in only prokaryotes. The most informative feature is the appearance of stop codons within the presumed translation of homologous DNA sequence. But SPURIOUS has dependency on ANTIFAM for training dataset. And as a machine learning based tool, SPURIOUS didn't get much good number of training data from ANTIFAM, and thus it is still in its early stage of development, and its results are considered as indicative, but they require inspection for most applications. The identification of spurious genes is an area of genomic annotation that has received very little attention. This is partly due to the difficulty of proving that a gene is not expressed in any condition. So this present study is an attempt to explore those possibilities, to find some ways of enriching ANTIFAM entries with confident and enabling future improvements of SPURIOUS. So for this, we have started searching spurious proteins with SPURIOUS 1.1, but even after having a high spurious score, it seems too difficult to be sure about that protein's spurious status. So we had to find out another way for becoming more confident on our decision. Then we came up with this BLASTX method, and if we look at this figure 1, we took a query protein, and as there is no way to do a protein versus protein search in different DNA reading frames, we had to make a DNA version of that protein, and then we carried out a BLASTX search against a real protein database, SwissProt. So if we find the negative any matches with this negative that is the minus frames, -1, -2 or -3 frames, this is the real protein, this is -1 frame protein, and this makes our protein on +1 frame a fake or spurious protein. This is a very simple concept but very effective, and eventually the idea of using U-search came on our mind because its swiftness and advantage over BLASTX in finding the overlaps. So this method has reduced the time and labor, but most importantly, it gave us the confidence in decision making of a protein's spurious status. So this new approach was much faster, and we could add around exactly 164 new families in spurious proteins from the shadow ops within only few days. This approach can be used as a new separate quality control system for genome annotations, obviously, and it will be strong candidate for being added as a new feature for Spureo to give its prediction much more confidence as well. And if we look at figure 2, this is clearly seen that ANTIFAM 6 has almost 3.5 times more entries and 3 times higher spurious like proteins in tremble matches found. So, and also we just searched for some IUPRET2 analysis or the protein disorder analysis. So this can surely show us that it gives a score 0 to 1, and for each amino acid of a protein, where 0 denotes no disorder and 1 denotes highly disorder. And we created a histogram with this average IUPRET value of real proteins and spurious proteins. We can understand very easily that the sequences having IUPRET average score greater than 0.45, these proteins are higher chance of being spurious. So we'll suggest that Spureo can be trained with a large number of spurious protein using our new ANTIFAM entries, and we can suggest 2 new features, that is BLASTX or use search analysis and the protein sequence disorder analysis. And we can expect that these steps will play a vital role in development of a powerful tool for identifying spurious proteins in a greater extent and making the protein database free from misprediction in near future. Thank you so much.